Welcome to Biotanemy. Electron Transport Chain. In our previous videos, we've followed glucose through glycolysis and the citric acid cycle. Glycolysis converted one glucose molecule into two pyruvate molecules, producing a net gain of 2 ATP and 2 NADH. Those pyruvate molecules entered the mitochondria, where the pyruvate dehydrogenase complex converted them into acetyl-CoA, generating another 2 NADH.
Next, each acetyl-CoA entered the citric acid cycle, producing 3 NADH, 1 FADH₂, and 1 GTP per cycle.
Since one glucose produces 2 acetyl-CoA molecules, the TCA cycle generates a total of 6 NADH, 2 FADH₂, and 2 GTP. ATP is the primary molecule that stores and transfers usable energy within the cell, and is therefore often called the energy currency of the cell. Although glycolysis and the citric acid cycle produce ATP directly, they generate much more energy in the form of GTP, NADH, and FADH₂. Since most cellular processes require ATP as the immediate source of energy, the energy stored in these molecules must be converted into ATP. But here's an important question. If NADH and FADH₂ already contain so much stored energy, why can't the cell simply use that energy to make ATP directly? The reason is that releasing all of that energy at once would be highly inefficient and could even damage the cell. Instead, the cell extracts the energy carried by NADH and FADH₂ in a series of small, controlled steps, capturing it efficiently to produce ATP. This controlled process takes place in the electron transport chain, or ETC. The NADH produced during glycolysis cannot cross the inner mitochondrial membrane directly. Instead, its electrons are transferred into the mitochondria through the malate aspartate shuttle or the glycerol 3-phosphate shuttle. In contrast, the NADH and FADH₂ generated during pyruvate oxidation and the citric acid cycle are already present in the mitochondrial matrix and can directly participate in the ETC. The electron transport chain consists of 5 large protein complexes embedded in the inner mitochondrial membrane. They are: Complex 1, also known as ubiquinone oxidoreductase or NADH dehydrogenase. It accepts electrons from NADH and pumps protons. Complex 2, or succinate dehydrogenase, accepts electrons from FADH₂, but does not pump protons. Complex 3, called the Cytochrome BC1 complex, uses coenzyme Q and Cytochrome C oxidoreductase to transfer electrons to Cytochrome C, and pumps protons. Complex 4 or Cytochrome C oxidase will transfer electrons to oxygen, forming water, and pumps protons. And Complex 5, also called as ATP synthase or F₀F₁-ATP synthase, uses the proton gradient to synthesize ATP.
What exactly happens inside the ETC that allows the cell to convert the energy stored in NADH and FADH₂ into ATP? Let's see. The ETC begins at complex 1. NADH carries one hydride ion, which consists of 2 electrons and 1 proton. Inside complex 1, NADH transfers this hydride ion to FMN or flavin mononucleotide, the first electron acceptor of complex 1. However, FMNH2 cannot hold these electrons indefinitely. It transfers them one at a time to a series of iron-sulfur clusters, acting as a bridge between the 2 electron donor, NADH, and the 1 electron carriers, the iron-sulfur clusters. The iron-sulfur clusters then transfer the electrons to coenzyme Q or ubiquinone, releasing a small amount of energy at each step. Complex 1 harnesses this energy to pump 4 protons from the mitochondrial matrix into the intermembrane space. Unlike NADH, FADH₂ does not pump protons into the intermembrane space, because the energy released during electron transfer is insufficient to drive proton pumping. Instead, it enters the ETC through complex 2, also known as succinate dehydrogenase, the same enzyme that catalyzes the conversion of succinate to fumarate in the citric acid cycle, making it the only enzyme that participates in both the TCA cycle and the electron transport chain. Unlike complex 1, FAD is tightly bound to complex 2 as a prosthetic group. During the oxidation of succinate to fumarate in the TCA cycle, FAD accepts 2 hydrogen atoms and is reduced to FADH₂. Rather than diffusing away, the bound FADH₂ immediately transfers its electrons through a series of iron-sulfur clusters within complex 2. The electrons are also then transferred to ubiquinone, reducing it to ubiquinol, which carries the electrons to complex 3. After ubiquinol binds to the QO site, outer Q binding site, of complex 3, it donates its 2 electrons and releases 2 protons into the intermembrane space. These protons contribute directly to the proton gradient across the inner mitochondrial membrane. The 2 electrons then follow different pathways. The first electron is accepted by the RISC iron-sulfur protein, which acts as an electron carrier within complex 3. The RISC iron-sulfur protein transfers this electron to cytochrome C1, another electron carrier embedded in complex 3. Cytochrome C1 then passes the electron to cytochrome C, a small, water-soluble protein located on the outer surface of the inner mitochondrial membrane. Cytochrome C can carry only 1 electron at a time, so it accepts this first electron and transports it to complex 4. The second electron cannot be transferred to cytochrome C because it already carries 1 electron. Instead, complex 3 directs the second electron through cytochrome Bl and then cytochrome BH, 2 heme-containing proteins within the complex. This electron finally reaches the Chi site or inner Q binding site, where an oxidized ubiquinone is already bound. After accepting this single electron, the ubiquinone is converted into a semiquinone radical. The semiquinone is only partially reduced because it has received 1 electron instead of 2. It remains bound at the Chi site, waiting for another electron to complete its reduction. When another ubiquinol arrives at the QO site, the same process occurs again. The first electron is transferred to another cytochrome C molecule and carried to complex 4, while the second electron follows the cytochrome B pathway to the QI site. There, it combines with the previously formed semiquinone radical and 2 protons taken up from the mitochondrial matrix, converting it into ubiquinol once again. This regenerated ubiquinol then rejoins the coenzyme Q pool and can participate in another cycle. The purpose of the Q cycle is to efficiently transfer electrons from ubiquinol, which carries 2 electrons, to cytochrome C, which can carry only 1 electron at a time, while simultaneously contributing to the proton gradient required for ATP synthesis. The reduced cytochrome C carries a single electron from complex 3 to complex 4, or cytochrome C oxidase, the final complex of the ETC. Now, the electrons cannot remain inside the ETC forever. Unless they are removed continuously, electron flow would stop, bringing ATP production to a halt. This is where oxygen comes into the picture. Oxygen, which diffuses into our cells during respiration, acts as the final electron acceptor of the ETC. Complex 4 transfers the electrons received from cytochrome C to oxygen. Since one oxygen molecule requires 4 electrons to be completely reduced, 4 cytochrome C molecules, each carrying 1 electron, successively donate their electrons to complex 4. Within complex 4, the electrons are transferred through 2 copper centers, copper A and copper B, and 2 heme groups, heme A and heme A3. These metal centers safely channel the electrons to oxygen while preventing the formation of harmful reactive oxygen species. Once oxygen receives 4 electrons and combines with 4 protons from the mitochondrial matrix, it is reduced to form 2 molecules of water. The energy released during this electron transfer enables complex 4 to pump 2 additional protons from the mitochondrial matrix into the intermembrane space, further strengthening the proton gradient. By the end of the electron transport chain, complexes 1, 3, and 4 have pumped a large number of protons from the mitochondrial matrix into the intermembrane space. As a result, the intermembrane space now contains a high concentration of protons, while the mitochondrial matrix has a lower proton concentration, creating a gradient difference between the 2 spaces. This difference in proton concentration creates an electrochemical gradient, also known as the proton motive force. Like water stored behind a dam, these accumulated protons possess potential energy and naturally tend to flow back into the mitochondrial matrix. However, the inner mitochondrial membrane is impermeable to protons, preventing them from diffusing back directly. The only route available is through complex 5, also known as ADP synthase or the F₀F₁-ATP synthase. Let's see the structure of ADP synthase molecule. It is a large molecular machine embedded in the inner mitochondrial membrane. It consists of two major functional regions, the F₀ region and the F₁ region. The F₀ region is embedded within the inner mitochondrial membrane and forms the proton channel. It is composed of the A subunit, the serine, and associated structural proteins. Its primary role is to allow protons to flow from the intermembrane space back into the mitochondrial matrix. Attached to the F₀ region is the F₁ region, which projects into the mitochondrial matrix. This is the catalytic head responsible for ADP synthesis. The F₁ region consists of 3 alpha subunits and 3 beta subunits, arranged alternately in a hexameric ring. At the center of this ring lies the gamma shaft, along with the delta and epsilon subunits, while a peripheral stalk anchors the catalytic head and prevents it from rotating. The beta subunits contain the catalytic sites where ADP is synthesized, whereas the alpha subunits mainly bind nucleotides and provide structural support. The F₀ is composed of 3 subunits A, B, and C. As protons enter the subunit of the F₀ region, they bind to an aspartate residue on a C subunit. This binding causes the serine to rotate. As the ring completes almost one full rotation, the proton reaches another channel in the A subunit and is released into the mitochondrial matrix. The aspartate residue is then ready to bind another proton, and the cycle repeats. The 2 B subunits act like a stationary support, holding the F₁ region in place while the serine and central stalk rotate to drive ADP synthesis. Because the gamma shaft is firmly attached to the serine, the rotation of the serine causes the gamma shaft to rotate as well. While the gamma shaft rotates, the surrounding alpha-3 beta-3 catalytic head is held stationary by the peripheral stalk. This difference in movement is the key to ADP synthesis. Instead of the entire enzyme spinning, only the central shaft rotates, causing mechanical changes in the beta subunits. Initially, each beta subunit is already in a different shape. Let's take the initial conformation as beta-1 is open, beta-2 is loose, and beta-3 be tight. They are not identical because the gamma shaft is asymmetrical, like an off-center camshaft inside an engine. In the open state, the catalytic pocket is open. ADP has very little affinity. Therefore, ADP leaves, making space for ADP and inorganic phosphate to enter. In the loose state, the pocket becomes narrower. ADP and inorganic phosphate are trapped. But they are still separate. And in the tight state, it squeezes the beta subunit even more. The catalytic pocket becomes very compact, forcing ADP and inorganic phosphate very close together. Because they are held in the correct orientation, they combine to form ADP. Hence, it is known as the binding change mechanism. ADP forms because the protein changes shape. Each 120 degrees rotation of the gamma subunit induces a conformational change in one beta subunit. After a complete 360 degrees rotation, produce 3 ADP. In mammalian mitochondria, the serine contains 8 C subunits. Each proton binds to one C subunit, causing the ring to rotate by 360 by 8 is equal to 45 degree. So, 1H equals 45 degrees rotation. Then, 120 degrees rotation requires 120 by 45 is equal to 2.67 protons. Since you can't have 0.67 of a proton, this is an average value over many rotations. Therefore, approximately 2.7H are required for one 120 degrees rotation of the gamma shaft. And approximately 8H are required for one complete 360 degrees rotation. Also, one 360 degrees rotation produces 3 ADP. So you can say, in mammalian mitochondria, approximately 2.7 protons are required to rotate the gamma shaft by 120 degrees, resulting in the synthesis of one ADP molecule. A complete 360 degrees rotation requires about 8 protons and produces 3 ADP molecules. The movement of protons through ADP synthase, driven by the proton motive force, is known as chemiosmosis. The coupling of electron transport with ADP synthesis through this proton gradient is called oxidative phosphorylation.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.